Good afternoon all. So in today's session we can discuss applications, then advantages, disadvantages of fluorescent microscope. So in the last session we have discussed different parts, working principle of fluorescent microscope. So according to these applications, this fluorescent microscope, it is used to identify structures in fixed and live biological samples. And also it is a common tool for today's life science research because it allows the use of multicolor staining, labeling of structures within the cells, and the measurement of the physiological state of a cell. Next, about advantages of fluorescent microscope. So this is the most popular method for studying dynamic behavior, which is exhibited in live cell imaging. So it helps to study the dynamic behavior of a cell. This stems from its ability to isolate individual proteins with a high degree of specificity amidst non-fluorescing material. So it helps to isolate individual proteins. The sensitivity is high enough. It is highly sensitive enough to detect as few as 50 molecules per cubic micrometer. Also, different molecules can now be stained with different colors, allowing the multiple types of the molecule to be tracked simultaneously. Next, these factors combine to give fluorescent microscopy a clear advantage over other optical imaging techniques for both in vitro and in vivo imaging. So what about limitations of fluorescent microscope? The fluorophores, they lose their ability to fluoresce as they are illuminated in a process called photobleaching. This photobleaching occurs as the fluorescent molecules accumulate. Then chemical damage from the electrons exhibit this type of fluorescence. Then another limitation, the cells are susceptible to phototoxicity, particularly with short wavelength light. So most of the cells get died. Furthermore, fluorescent molecules have a tendency to generate reactive chemical species when under illumination, which enhances the phototoxic effect. Unlike the transmitted and reflected light microscopy techniques, fluorescent microscopy only allows observation of specific structures which have been labeled for fluorescence. So that's all about fluorescent microscopy. Now next is interference microscopy. It can be of differential, interference, contrast microscopy. Here we have given differential, interference, contrast microscope configuration. If you want to, okay. Then introduction. Over the past few years, the microscopic techniques had faced with some complications while observing the live specimens. And the important complications are transparency, light, and because of that because of this the image produced doesn't have any sufficient contrast. That is the problem. So thus with the development of this interference microscopic techniques, it was possible to now visualize specimens with adequate contrast. Next is about principle. Interference microscopy uses a prism. So what is the principle? A prism is used, and when the light passes through this prism, it splits the light into two slightly diverging beams. So two light rays move towards each other opposite side. They diverge while passing through the and then while passing through this prism, the light splits and this into two diverging beams, and then they pass through the specimen. This diverged beam of light now falls on the specimen. It is thus because of the measuring the difference in refractive index upon recombining the two beams. So now we can measure the difference in the refractive index upon these two beams which already have diverged, and we can measure the difference in the refractive index of that diverged beams. Upon how? By recombining the two beams. Now this interference occurs when the light beam is retarded or advanced relative to another. That is the interference. That is the main principle. So the interference microscopy or quantitative interference microscopy, it is one of those techniques, and it has been derived from phase contrast microscopy, but it is more sensitive than this technique and make possible the easy and clarify viewing of living organisms. This technique is used by taking light. How? Now how this technique goes? Working principle that the light is taken from the condenser and using a prism, it separates the light into two beams. One beam is called object beam. That object beam which goes through the object such as specimen and the objective lens. Other beam is called reference beam. It goes through another objective without touching the specimen. So object beam touch the specimen, reference beam doesn't touch the specimen, and these beams allow a specimen to be seen through the difference in the fields. So one has passed through the specimen. So these two beams helps to see the specimens. How? Through the difference in the field which are caused by two beams, and also difference of the two images which are formed by these two beams, allowing the details to be seen. So this is the principle while passing through the prism. This type of image, this is the principle. So there is a prism is placed. Here first of all a polarizing filter is placed, and this prepare the light for the first Wollaston prism. And this is the Wollaston prism, the first prism placed. It separates incoming light into perpendicular polarized components. Now then passing through the condenser lens, then sample, then objective lens, then Wollaston prism 2. And this recombines the, so already separated beams, they recombine in this prism. First prism they got separated, and in second prism they combine. Combining the incoming perpendicular polarized light into same polarization. Then again there is another polarizing filter placed on the path of the light. Now this microscope is a bright field light microscope. Addition of following elements, that is a polarizer between the light source and condenser. Is there any polarizer? Yes. This is the polarizing filter. Then a polarizer between the light source and condenser. Then DIC beam splitting prism, DIC beam combining prism, and analyzer. Manipulating the prism changes manipulating the prism changes the beam separation, which alters what? The contrast of the image. When two beams, both reference and object beam, when the two beams pass through the same material across the specimen, they produce no interference. But when the two beams pass through different material across the specimen, such as on the edges, they produce the alterations when combined. Next is the different types of interference microscope. There are mainly three types of interference microscopy, that is classical interference microscopy, differential contrast interference microscopy, Then fluorescent contrast. Since its introduction in late 1960s, this differential interference contrast microscopy, it has been popular with it has become popular in biomedical research because it produces high resolution images. This interference microscopy produces high resolution images of fine structures by how enhancing the contrasted interfaces. So now the image produced is of a thin optical specimen section and appears three-dimensional with a shadow allowing it. Then this creates a contrast across the specimen, which is bright on one side and darker on other side. And also there is a variation in this interference microscopy called differential interference contrast microscopy. This is also known as Nomadsky interference contrast microscopy. The optical microscopy illumination techniques used to enhance the contrast in unstained or transparent samples was named after its inventor and also uses two beams produced by the single polarized light. Initially, the polarized light is divided into two rays polarized to each other, sample and reference. So, polarized light divides two rays polarized to each other, and this enters the first Nomadsky modified Wollaston prism. Then these two rays are focused by the condenser for the passage through the sample and then travel to the adjacent areas of the sample. Then divided by the shear. That is, separation is normally similar to resolution of the microscope. So this is the picture again I am showing. Wollaston prism. Two types of prisms are there. These interference techniques have advantages. What are the advantages in involving living or unstained biological samples can be easily observed, and their application in biology, crystallography, mineralogy and chemistry. So they have in these fields also this microscope have more advantages. In standard optical microscopy techniques, its resolution and clarity is also visible. So this can be used, this have important applications in biology, crystallography, chemistry, etc. After that, they will face different optical wavelength where areas differ in refractive index or thickness will cause a change in phase of one ray relative to another, according to the delay experienced by the wave in more optically dense materials. Lastly, the rays goes to the objective lens and are focused for the second Nomadsky modified Wollaston prism, which join the two rays into one polarized light, and this make an image with a three-dimensional appearance. This final junction of rays lead to interference by brightening or darkening the image at that point according to the optical wave difference. Then fluorescence differential. Next is about, already I have discussed differential interference contrast microscopy. Next is fluorescent differential interference contrast microscopy. It was developed by combining fluorescent microscopy with the DIC to minimize the effect of photo bleaching on the fluorochromes bound to the staining specimens. The same specimen is equipped to simultaneously image a specimen using DIC and fluorescence illumination. So that then next is about limitations. These techniques have their own limitations as its requirement for a transparent sample of similar refractive index. It is quite difficult. And it is inadequate for thick samples, only useful for thin samples and for the most non-biological samples. These are not useful for the most non-biological samples because of its polarization dependence. So that's all about interference microscope. Thank you all.